कैरेक्टर अलग हो जाता है। मैं इशारा आगे का नहीं कुछ कर रहा हूँ, ऐसा मत समझिएगा। तो थैंक यू वेरी मच, और सर को बहुत बहुत धन्यवाद पटना की ओर से, बिहार की ओर से, टीपीएस कॉलेज परिवार की ओर से।
Titled Recent Advancements in Basic and Applied Material Sciences. Today we gather to share our knowledge, expertise and innovations in the field of material science, and I am honored to be part of it. I am Shwetanshu Mala, Professor of Physics, Patna University, and I will be chairing the session starting 4 to perhaps 5 p.m. There will be 2 talks in this session. Every speaker will have 25 minutes for presentation and 5 minutes for questions and discussions. Dr. Pratyush Kumar Mishra from Oxford University, UK will be the first speaker who will be enlightening us with his talk titled Chemogenetic Platforms for Live Cell Imaging, Sensing and Interaction Mapping. Pratyush Mishra, currently a research associate at the University of Oxford and the Rosalind Franklin Institute under the Wellcome Trust Discovery Grant. He earned his master from IISER Pune and moved to earn PhD in chemistry from UNIST South Korea, where he developed novel photo-cross linkers and proteomic tools for studying molecular interactions in live cells. His postdoctoral research at Seoul National University further advanced proximity leveling chemistries and enzymatic probes for spatial proteomics. Dr. Pratyush Mishra's work focuses on chemogenetic tool development, chemical biology, and proteomics with application in understanding bacterial drug resistance and cellular signaling. He has received multiple awards, including the Chemical Science Oral Presentation Prize, Royal Society of Chemistry, and several Best Poster Awards at different international conferences. With a strong publication record in high impact factor journals such as Nature Communications, Nature Chemical Biology, ACS Central Science, Dr. Mishra continues to pioneer innovative approaches to probing dynamic molecular networks in biology. Now, Mr. Mishra, the floor is yours. We are waiting for your enlightening talk.
Thank you very much, Professor, for the opportunity to present my work. So, as Professor mentioned, I started working on chemogenetic tools to study cellular architecture and interactions. So, why we need to study cellular architecture and interactome? So, there are protein transcripts, DNA architecture, and cellular networks. Overall study of all these in a spatial manner, where what is located and how it interacts with the other entity, is called spatiomics. So as a group, several scientists came together and decided to study this kind of work. For this, we needed to develop some tools which can identify the population residing in a specific location. For that, we developed proximity labeling tools. So for proximity labeling, to enlighten everyone how it works. Let's say this is the area on which we want to look at what is there. We introduce an enzyme which is marked in yellow, and this enzyme can be seen as a spray can which can emit any activated probe to label everything nearby. And generally this is tagged with a biotin probe, and we can see this probe enriching nearby proteins and enlightening us what is in this small square box. And this box is known as proximity labeling radius, which is counted from the enzyme tag. So depending on how large and small is the proximity labeling radius, we get the idea of everything around that tagged enzyme. So first enzyme developed in this field was Bure. So what Bure does is it takes a specific set of peptide with a specific sequence. It utilizes biotin plus ATP, and using this ATP and biotin, it makes biotin AMP, which biotin AMP can attack the lysine on a specific peptide, giving us the tagged peptide. And this biotin is very important. It can be enriched and later can be identified mass spectroscopy. However, it causes a problem because this Bure enzyme is selective to this particular peptide. Several groups came together and decided to mimic this Bure and make several mutants of it. One of those mutants, Bure R118G, cannot distinguish any peptide from the sequence. So, when you provide biotin with the ATP, without differentiating any peptide, it labels all the peptides uniformly, giving us the information of all the lysines available in that region. However, this Bure was very big protein. Sometimes it causes problems in the cellular localization of the protein we are interested in. So people came together again and decided to modify the N terminus, C terminus, shortening the C terminus, or taking this same enzyme from another species. And gradually they started to reduce the size of it, and from 35 kilodalton up to 28 kilodalton they have successfully utilized. This is a brief history of the development. This is the original Bure from which R118 was created in 2004. It took almost 8 years to make new BioID, and then 4 years to make BioID2, and then development accelerated, and recently we developed several turbo ID which requires very little time, that's why turbo, and then we have micro ID which has very short labeling radius. However, all these enzymes require biotin as a substrate. However, as a scientist, we all see that biotin has large scope. However, sometimes it is not possible to utilize biotin owing to its solubility and its natural localization. So people are still working on finding a new enzyme which can accommodate any other probe which is not biotin. So this is the focus. This part of the talk was focused on tagging the lysine within a labeling radius. However, this is not the only enzyme. There is also HRP, horseradish peroxidase, which is traditionally used in cryo-EM imaging or imaging using the osmium tetroxide. This enzyme had several disulfide linkage which are very much important for the folding of this protein and retaining the activity. However, in some environments of the cell, let's say in ER, there is a high redox reaction, redox environment, and several redox reactions are going on. In those reductions, some of these cysteines can be broken into thiols, disulfide to thiols, and the secondary structure of protein is broken and rendering it useless. So overcome this, Alice Spring group from Stanford came forward to develop, instead of horseradish peroxidase, they have taken soybean ascorbic acid peroxidase, which also has the same heme moiety and it does not have any disulfide linkage, by which we can utilize it in high redox condition, making exactly same substrate and same applications. However, she noticed that when apex is tagged to one side of the membrane or another side of the membrane, depending on which side is exposed to apex, it can generate different kind of labeling, giving us information that this protein has C terminus on the outside or the inside. That helps us in identifying several proteins for which terminus information was not available. However, it was not only limited to that. People can also make half of the apex on protein A and half of the apex on protein B. When protein A and B are interacting, this complex can act as a molecular glue, and this active apex can be used to carry out the reaction. Using this, they have taken mitochondrial outer membrane tag FKBP and ER tag FRB. When these two proteins come together, they generate the apex and we get the signal. So this has been very useful tool to identify the organelle contact sites. And using this, you can see the mitochondria marked as M, and one side of the mitochondria is more contrast. That signifies it is in contact with the ER. So these kind of tools can be used to identify if mitochondria is interacting with the ER or not at that